All right, so our next chat today is from Dr. Heiner Wedemeyer, and he will be joining us online. As we all know, he is a leading hepatologist and with a great deal of expertise in both hepatitis B and hepatitis delta virus. And his talk will be on HBV and HDV cross-utility for drug development.
Okay, hopefully you can hear me.
Yes, we can.
Okay, very good. So I think the most frequently asked question of the last 18 months, can you hear me and can you see my slides? So these are the ones. Okay, thanks all of you for involving me. And I really appreciate this discussion. It's really great. And my task is basically to switch gears slightly and discuss if HDV could also help us to develop drugs against HBV or both. And I think this is really a very interesting idea, and therefore congratulations that you included this topic into this symposium. Brief talk, these are my conflicts of interest, including companies developing drugs against HBV and HDV. So why HDV? This is obviously the key question, why even to talk about this. Is a strange virus, it's a strange disease, it may only cause problems if you run clinical trials here, and it may harm your drug development. And I thought to briefly go through a SWOT analysis in this talk. So what could be strengths, weaknesses, opportunities, and threats to develop drugs in Delta, and this will be the analysis which I will discuss in more detail at the end after having shown you some of the data. So I think we can talk a lot about strength, opportunities, but there are certainly challenges and threats when you go into Delta for clinical trials. We also have to really differentiate when we talk about drug developments in Delta. Are we looking for novel drugs against HBV, which we want to study in HDV co-infection, or are we talking about drugs specifically dedicated to Delta, where we leave S antigen aside, and the aim is really to reduce Delta replication only, and the underlying HBV infection may be to be considered as a carrier stage with not causing much problems. So these are the issues behind this. So Delta, I think we always have to remind ourselves that this is, I think in biology, a cool system. So you have a pathogen and it's hijacking other membranes. I will not show you the data from Venezuela published earlier this year in Hepatology, where they screened, following the Nature.com paper from France, HCV patients without HBV markers for Delta, and identified 2 patients who were anti-HBV positive, 1 patient with RNA positivity for Delta, in complete absence of any HBV marker, at least serologically, which I think is interesting. So the phenomenon that Delta may hijack other membranes to disseminate is quite interesting. The other issue, obviously, with Delta is Delta does not encode for own viral enzymes, so it's challenging, or it's not possible to develop, let's say, enzyme inhibitors specifically dedicated to Delta. It uses host enzymes, but there have been screens already published, either from the Strasbourg group or also from Stefan Urban. And there may be hits that we have really also DAAs against Delta with limited effect of host proteins could be possible. When Stefan published this nice review in Gut earlier this year, and it really highlights that we have distinct steps in the HDV life cycle which could be targeted, for example, the farnesyl transferase, but there are also common effects which may be then also relevant for HBV infection. What is quite clear, and this has been shown in numerous papers, that Delta takes a more severe long-term course than HBV monoinfection in many, but not all patients. And that's important, I want to highlight, and I don't show you data, but there are Delta patients who do not show an aggressive course, who have, let's say, some level of fibrosis, and who could be potential candidates for a phase 2A study without taking the risk of going directly into patients with advanced liver cirrhosis. One challenge with Delta is the heterogeneity worldwide. We are talking about 8 different genotypes. We are talking about different subgenotypes, and that can be combined with different HBV genotypes. Again, Stefan Obam published in JHIP in April an in vitro study where he combined all HBV genotypes with all HDV genotypes, and overall everything is possible, but there are distinct differences when matching these viruses. What we also have to keep in mind, and Adam talked very nicely about HBV and immunity, that also immune control of Delta is possible. There is a lot of data already on some immune cells, but also from a clinical perspective, 30%, 20 to 40% of anti-HDV positive patients are HDV RNA negative. And there have been studies on innate immunity, immune cells in HDV. We did some studies on NK cells. Nicholas Bergström from the Karolinska Institute studied mate cells in HDV infection. There have been data from Freiburg and Munich on HDV-specific CD8 T-cell responses, where also immunoscape has been shown. And there's a very recent paper in Gastro by Barbara Rahman's group from Helene Kafalakis, who showed that not only virus-specific T-cells, but also liver-resident bystander CD8 T-cells may contribute to liver disease and the pathogenesis in HDV infection. And I think that's important also when we talk about stimulating immune responses to get rid of virus-infected cells. We also have to keep in mind that we may have collateral activation of bystander cells, which may be also a problem or a benefit, and that's what we need to understand, and therefore we really need to study the liver. So when we talk about Delta, obviously there are quite some data that have frequently been shown. We have the nucleic acid polymers, which have been discussed frequently with the spectacular S antigen declines. However, and that is the first challenge in Delta, in that paper, also these ALT flares have been shown, which were quite significant in some patients. And obviously, if a patient has liver cirrhosis, this may be a problem. The follow-up of that trial has been published in Hepatology Communications, showing very good long-term responses in a significant number of these patients. And also the analysis of the ALT flares were published in J-viral hepatitis, suggesting that they were following Grace's talk, were mainly beneficial flares. But still, we are talking here in Delta about cirrhotic patients, and these flares need to be followed with caution. You don't want to have this in an uncontrolled way. Then we have the Prenylation Inhibitor, which has been studied first by the NIH group and then by Chian-Udaidin. The phase 3 trial exploring lonafenib is currently ongoing, and it's still recruiting. It's a large trial with a couple of hundred patients, and where we should expect data maybe in 1 or 2 years. And that's certainly something where viral decline has been shown. And the drug has also been studied in combination with interferon lambda by Chris Ko at the liver disease section of the NIH. We have seen the data at the last liver meetings with significant declines, and also some patients with off-treatment responses. So, and again, this is something to be explored. And then obviously we talk about entry inhibition. Bulevirtide has been discussed frequently during all these recent meetings. Summarizing the data on bulevirtide, I think it's important to where we can learn a lot from HBV pathogenesis. So having used this compound in Delta, we learned also something about B. So first of all, yes, we saw this HDV RNA decline when using bulevirtide alone. Maybe a small dose response, but not very spectacular. But, and you're all aware of this one, nothing happened on S antigen levels. And I think this is really something we should talk about. So you protect not yet infected cells with an entry inhibitor, and your HDV RNA is declining, so possibly losing infected cells. But S antigen remains stable, and is the explanation here that the S antigen is produced by integrates into the human genome in cells not carrying RNA for Delta anymore, to be discussed. On the other hand, then the very interesting observation that when you combine the compound with interferon, that there was a significant S antigen decline in the low dose bulevirtide group, but not in the high dose bulevirtide group. And then the question is really, could this be because here we see higher increases in bile acids, that this somehow interferes with interferon effects? And then when we then talk about immune stimulation, what is the role of bile acids, which may vary in HBV infection when using the immune stimulators? And I think this is something that, in my view, has not been considered in many cases. And the drug is available in Europe. We can prescribe it here in several European countries. So now, finally, what are then the hurdles and opportunities for future therapeutic options? So let's talk about the strengths. The good thing in Delta is we have really, meanwhile, standardized virological endpoints. We have HDV RNA, we have another biomarker, HDV RNA, reflecting number of infected cells. We can do liver biopsies. It's ethically easily possible because we don't know what's going on. For example, there may be also underlying autoimmunity, and patients tolerate this. And the patients are very motivated. So these are really opportunities. There are and strengths. And obviously the opportunities is Delta is a high unmet need. There is no fully approved therapy yet. Bulevirtide is only conditionally approved. And therefore, for new treatments, some side effects may be acceptable, as from some of the currently ongoing trials we know. And obviously, we could help HBV drugs, so there's an opportunity. The challenges are still that Delta is an orphan disease. So we don't have that many patients. I talked about the heterogeneity. The epidemiology of Delta is really strange. In some countries, we have very high prevalences and others low, and even in Europe, there are huge differences. The other point is there have been discussions with the agencies, what are really the surrogates for clinical endpoints. So is HDV RNA suppression always associated with an improved clinical long-term outcome? Similar discussions that we had in HCV are ongoing in Delta. And obviously for new drugs, you always have to compare to what is available, and that would be bulevirtide at this stage, which is a challenge for new drugs. And the threats, I mentioned already, is the severity of liver disease. We are talking about 2 infections. If you reduce 1 infection, the others may jump up, but depending on the mode of action. Also, relapses after treatment may be an issue. And we also talked about these late relapses after interferon therapy, which, for example, we just published in the Journal of Viral Hepatitis, the HIDD-1 trial, the long-term follow-up. We observed a relapse 7 years after initial response, so there's no SVR. The question then is, do we need backbone interferon for some patients? So that's certainly an issue. So what are then the clinical pathways? The patient populations for new trials in Delta would be, they must be positive for RNA. They should have compensated liver disease. And if I can include patients with cirrhosis, that's really to be discussed. It's not that easy. We have to be very careful. The clinical need is, however, in patients with advanced cirrhosis, and there are opportunities if you have safe drugs. The question then always when we talk about development pathways, dose finding in Delta, simply because of the number of patients available, is challenging. Therefore, it would be good to have an initial dose finding in HBV monoinfection and then go with a defined dose to Delta. And there could be different designs. The question is also in the phase 2b and 3 whether you have to test it, can still test it versus placebo, or whether you have to test this against bulevirtide. The duration of treatment then required in Delta is also an issue. There we may get some ideas from the ongoing bulevirtide trials where treatment for 2 years, where the 3 years of treatment is currently tested. And the key question in Delta is really whether we need long-term suppression, like with the NUTSIB right now, with maintenance treatment, or whether a finite treatment is possible. When we talk about endpoints, I talked about HDV RNA, we have biochemical endpoints, we still can biopsy our patients, and obviously the biomarkers that Massimo nicely introduced should also then apply for Delta, and then we can talk about the utility of these markers in Delta. There have been some discussion on the surrogate markers for treatment efficacy. I want to refer to that paper and not go too much into detail where we discussed all these different markers and the usefulness in Delta. My last slide, so what have we learned from previous trials and data in Delta? I think the opportunity is that we really had fast recruitment in countries like Russia and Romania. So it's possible, the patients are there, you have to go to the right sites in the right countries, and then you can really run trials quickly. We have seen ALT flares, we discussed this, and in Delta they can be severe, and that has to be followed very closely. Patient-reported outcomes should be studied in all phases, in particular in Delta, and that is something we have not discussed this morning, which is an opportunity. And we also have learned that even though biopsies are possible, they really showed a huge variability, which is to me a challenge when you go for fibrosis as an endpoint. And I don't want to discuss different ideas for maintenance, finite pulse therapies, combinations with interferon. We may discuss if needed. And the last slide I always show is that coffee is good for the liver, and this also holds true for Delta patients. Thank you very much.
All right.